I'm going to take a slightly different view of breast cancer and really talk about where we are and why we are going in the direction that we're going. But I wanted to start out by saying that perhaps the biggest idea that we're going to be discussing is the idea that was initiated by NBCC, and the idea that we have to work together, we have to get out of our comfort zone, we have to set goals for ourselves that we aspire to achieve, and without that type of directive, all of the details of what we're doing as individuals aren't going to address the compelling and overwhelming demand for action and results. So again, to me, the big idea is exactly what we're doing today, exactly what NBCC and the leadership have driven us to do to make that kind of a difference, really change the status quo. So let me talk a little bit about our paradigms of cancer and why we manage cancer and why we screen and treat precursors for cancer. And it relates to this idea that there are elements of the breast cancer continuum and all cancer continuum in which intervention can alter that trajectory. So a recent review showed this illustration in which exposure leads to cancer, and people conceptually don't accept that, and they feel that it's more likely that exposure leads to a precancerous condition, and in additional exposures, additional genetic changes, the spell checker not working and so forth, all those elements eventually lead to the cancers that are then lethal. And so time allows for this accumulation, and time, exposures, and so forth. Much of that in the past decade it came from twin studies in which identical twins were followed in Scandinavia, and the concurrence rate, if you had an identical twin that had breast cancer, the rate of the identical twin getting breast cancer was only about 15%. So the sense that it wasn't all genetics, there were events that were occurring that led to cancer. So what are the best examples of that? And we've heard a little bit about cervical cancer. You know, at the conceptual level, we think that you have a normal cervix, there's an exposure to HPV. Many women get exposed to HPV, many women clear HPV, many women have chronic HPV infection, and still that becomes something that's indolent. But there is a progression to a lesion, a precancer lesion that's more accepted as precancerous, and that leads to cancer. So the idea that the precancer 100% of the time has to lead to cancer, it's not clear that that has to be absolute. It has to be a general propensity, a trend toward developing cancer, and then we accept the fact that it's a precancer. You have a great increased risk of developing cancer when you have that precancer. And clinically, at least in cervical cancer, we recognize that. And this is a cartoon on your left showing the normal architecture of the surface of the cervix. And then going to your right, there's a dysplasia probably related to infection, and then the genetic changes associated with infection, and then progressing on to invasive carcinoma. But right before invasive carcinoma, there's a condition called in situ carcinoma. And again, that is the clearly accepted precursor. And then severe dysplasia, moderate dysplasia, and so forth all exist. So this continuum is the kind of the paradigm. Now, what are the consequences of accepting that paradigm and accepting the fact that there's precursor, precancers, if you will? Well, the reality is that an action on that concept should lead to benefit to the population. And what I've shown you here is this is a picture of Eva Perón, who died in her 30s, in the 50s, from metastatic cervical cancer. Again, probably one of the most prominent and powerful women in the world who died. And what I show to the right is the incidence of cervical cancer, and the mortality is actually associated with this decrease in incidence. So the implementation of cervical cancer screening and other strategies managing the precursor lesion has dramatically dropped the mortality rate and the incidence of cervical cancer about 85% as far as reduction in deaths in the United States. Cervical cancer as a worldwide problem, the identification of this precancerous lesion and the screening and appropriate management has led to reduction in incidence, and again, we're hoping to see that data about mortality. Well, what about breast cancer? Well, breast cancer perhaps is more complicated and maybe not as unified in its etiology as cervical cancer, and genetic tests are being developed describing a variety of molecular subtypes. We are sort of utilizing technologies that were originated 100 years ago in screening mammography. And we use the anatomic distortion associated with the passage of an X-ray beam as the fundamental technology in mammography, and we improve it a little bit. We have better sensors, and we can use computer-aided diagnosis and so forth. And you can see on the left there, the mammogram is more clear than on the right, digital versus conventional. But still, the fundamental technology is the same. We're looking for this anatomic distortion. And despite the implementation of widespread screening, you can see in this chart there's an increase in the incidence of mammographic screening. And you can see a significant increase in the detection of ductal carcinoma in situ, what is felt to be the precursor for breast cancer. There hasn't really been the reduction in incidence of breast cancer. And as you've heard, there might be a small reduction in the mortality, but not the dramatic 85% reduction in mortality that we'd seen with the management of cervical cancer. So the question that we have is, why is that? Are we not identifying the appropriate precursor lesion for breast cancer? Are we assuming that it's ductal carcinoma in situ or something else in the breast? Or are we assuming that we're detecting the precursor by mammography, but perhaps we're not? So, as I mentioned, ductal carcinoma is thought to be the precursor. The most common form is ductal carcinoma in situ. And in this cartoon, you see the architecture. I think you've heard some talks earlier this morning. Again, the very beautiful illustrations provided earlier showing that the carcinoma cells within a duct don't disrupt the duct, and the mortality can be very low because there's a very rare chance of metastases in ductal carcinoma. But it does lead to invasive carcinoma. And so what I would say is we've heard that ductal carcinoma is the precursor. We've seen a rapid increase in the number of cases of ductal carcinoma being treated. We haven't changed our treatment of ductal carcinoma in situ. But if you look at the National Cancer Institute and their attempts to create a genomic map of breast cancer, they've really focused on relatively advanced breast cancers. They're identifying cancers that are 200 milligrams. They have to be 80% epithelial cells. We heard from Peter that 60% of cancers are mostly stroma, but the NCI is focusing on epithelial stroma-free cancers. The cancers in the NCIs evaluated, most of them are stage 2 and 3. But if we'd looked at the Duke Tumor Registry for the year 2011, a very large academic medical center, 20% of the patients coming in with a diagnosis of breast cancer have ductal carcinoma in situ. And the number of cases that the National Cancer Institute has included in the Cancer Genome Atlas is exactly 0.0%. So are we not focusing our lens on the dramatically increasing cohort of patients that have ductal carcinoma? Are we not focusing perhaps some interest in the precursor lesion that could be very actionable and lead to new prevention strategies, et cetera? So again, this is the question we have, and one of the things that we learned is we have to go to the literature and look for that, and have others have looked at the carcinoma in situ. And people have done some studies. This is a study of 25 cases of ductal carcinoma in situ. They used gene expression analysis to try to compare ductal carcinoma in situ to invasive carcinoma, the 1,000 genomes that the NCI has. And what they found was that ductal carcinoma in situ was identical to invasive carcinoma. So again, it's a very small data set, but how could indolent disease. You've heard that maybe some doesn't have to be treated. It's a very highly curable, but at the genetic level, which we've all learned is the fundamental wiring of the cell, at the genetic level, it's identical to invasive carcinoma. Again, this calls into question, perhaps we're ignoring the stroma. What are the inflammatory changes? What are the actionable elements in the breast that are occurring that are making a difference in the natural history of ductal carcinoma in situ? So as I said, one of the big lessons that I learned, again, in sort of taking a page from NBCC, is that we can't do this alone. We have to address fundamental questions that are overarching by building a team and building a consortium and working together. And this is what we're trying to do in an early breast cancer consortium, to really focus our lens on these potential early precursors. I'm not saying we know what they are. I'm not saying we've found them, but I think if we don't look for them, we'll never find them. And so one of the challenges is how do we identify these early breast cancer precursors? And this is just some text that we're happy to talk about, but fundamentally I want to go to this issue that what we know in the earliest stages of cancer is that there are molecular events that are occurring, metabolic or signaling, all of these events that are being well described by scientists around the world. But as we look at the strategies for detecting breast cancer, we still rely on anatomic change or anatomic distortion. So the biochemical processes are occurring, but we're waiting for the cells to grow to such a large volume that they distort or they cause microcalcifications. So this is a slide that shows that our detection method allows us to detect clinical tumors of about 1 gram. This is sort of 30 doublings. If we use mammography, the best technology that exists today that's proven to have an effect, we can improve detection to 27 doublings. Not a big increase. The molecular changes occurring within the tissue are probably occurring throughout this continuum. And the challenge we have and the opportunity we have is to actually detect these oncogenic signalings much before the anatomic change. And so the anatomic signaling, those characteristics that are the hallmarks of cancer, they are occurring. We've heard elegant discussions about metabolic changes, receptor tyrosine kinases like HER2, EGFR, mTOR, all of those are occurring in the breast. Why can't we detect them before they grow so big that a mammogram that can detect a broken bone can detect them? And so again, our focus is to look at molecular changes within the breast. We focused on a chaperone protein. Again, this cartoon here shows that we can actually identify these chaperone proteins by their chemical binding, and we can link these chemicals to probes that we can now identify. And this leads to this issue that we can make hybrid probes that are highly diffusible, can get into cells, and allow us to detect them. Well, you might have said, well, I've heard of this before, haven't we? Don't we use antibodies now and make them radioactive and don't those bind? This is what we're really talking about. We're now using chemicals that are hundreds of times, 1,000 times smaller than monoclonal antibodies that tend to stay in the circulation. These chemicals really diffuse in and allow us to bind into cells. And again, this shows you that an antibody relative would be the size of an 8-year-old boy, and a chemical would be the size of a marble in his pocket. So that marble is going to be able to get all over the house where the boy is going to be contained. So we're looking at these chemical moieties. This is some cartoons of how we imagine that they're working, and I'll tell you that we're very excited, and these are unpublished data, and I apologize for that. We tend not to usually publicly disclose things that are not peer-reviewed and published, but we're very excited that in this example we can see breast cancer cells not exposed to an antibody, but exposed to fluorescent drugs that take these drugs up and allow us to detect them. And I can tell you that in animals that have these cells in them, we can actually detect tumor cells taking up this drug and seeing fluorescent tumor cells much before we can feel or image a lump. So again, this is some in vivo data that I wanted to finish with that says we're very excited about the potential to use a variety of strategies, signaling pathways that are going to be discovered that we hadn't even thought about, but the conceptual framework of working together, developing strategies that identify the signaling pathways in cancer before the anatomic change occurs will help us at least characterize or perhaps identify these early precursor lesions of breast cancer. So we're very grateful to the CDMRP and this group of investigators, again, that represent a wide variety of scientists, classically not involved in breast cancer, pharmacology, neuroimaging, chemistry, optical physics, and so forth and so on. Again, the big idea here is the NBCC and its leadership and its ability to create coalitions, inspired us to really address these fundamental questions, try to build a team, and really have an impact. Again, I wanted to say thanks to all of you. I think the elements that you bring, the vision that you have brought, have allowed me to learn a lot from you. I was very inspired to hear about the project lead participants yesterday in the session we did, and I thought a lot about the accolades that were provided to scientists and clinicians about teaching the advocates in the project lead. But I want to tell you that we have learned a lot from you. We have learned and are inspired by all that you do, and I wanted to thank you for your leadership and everything you've done for all of us.